Okay, so welcome back to this next video in which we are discussing angiogenesis and angiotherapy. Okay, so we're currently in the process of discussing angiotherapy, and we're looking at examples of where you want to have an anti-angiogenic effect. Okay, and we've seen the cardinal example of an anti-angiogenic drug, which is bevacizumab, which is a monoclonal antibody that's been humanized, that is targeting a specific epitope of vascular endothelial growth factor A. And when the antibody molecule binds to the vascular endothelial growth factor A, basically it neutralizes the VEGF-A, and the VEGF-A becomes useless after that, so it no longer signals for the angiogenesis. Okay, and there was great hope that this was going to be able to cure all forms of cancer. So far it's been quite disappointing, and potentially the reason that it's been quite disappointing is because we're giving the drug once you've got very developed cancer. Okay, so the tumour has probably developed a blood vessel network by the time we're actually giving the drug, and that might explain why it's only having a modest improvement. But there is still a lot of research into targeting angiogenesis in the treatment of cancer. Okay, right. Now what we're going to see is another use for bevacizumab, which is also called Avastin, which is in the treatment of diabetic retinopathy, which is another example of a disease where you want to have an anti-angiogenic effect. Okay, so let me tell you about diabetic retinopathy. Okay, so the key symptoms then of diabetes mellitus are that you have hyperglycemia. Specifically, you have fasting hyperglycemia, which means that even when you have not just eaten a meal, you have elevated blood glucose levels above what it should normally be. Okay, so let me explain this. So normally, a healthy person who is not diabetic would have a blood glucose concentration of around 5 millimolar whilst fasting. Okay, so this is the normal fasting blood glucose concentration. Now if you're in the States, you don't like using the units of millimolar. Instead, the units that we use in the States are milligrams per deciliter. To convert between millimolar and milligrams per deciliter, all you have to do is multiply by a factor of 18. Okay, and if you like, you can try and work out, using your chemistry knowledge, why the factor is 18, okay, using the molecular weight of glucose. Okay, so all you have to do is multiply by 18 to convert between those two units. Okay, right, so normal fasting blood glucose concentration is 5 millimolar, which is 90 milligrams per deciliter. Okay, now, in people with poorly controlled diabetes mellitus, the fasting blood glucose concentration is above 7 millimolar, which is the equivalent of 126 milligrams per deciliter, again just multiplying by a factor of 18. Okay, so if you have a fasting blood glucose concentration of above 7 millimolar or 126 milligrams per deciliter, then that's the definition of diabetes mellitus, basically. Okay, in addition to having elevated fasting blood glucose concentration, i.e. fasting hyperglycemia, you also have absolutely massive postprandial glucose spikes. So let me just explain this. So if we draw a graph is the best way to do this. So on the y-axis, let's have glucose concentration in the blood, okay? And on the x-axis, let's have time. And then in a non-diabetic person, the blood glucose is usually at 5 millimolar down here, okay, or 90 milligrams per deciliter, okay, and then when you eat something, when you go into the non-fasting state, the fed state, then of course your blood glucose spikes up, okay, but then it comes back down quite quickly because of the effects of insulin, okay, and you have therefore a nicely controlled spike in your glucose concentration. Okay, so this is what's called a postprandial glucose spike, and these occur in everyone basically. When you eat, you get a postprandial, post meaning after, prandial meaning eating, postprandial glucose spike, or we'll just call it a postprandial spike. Now in people with diabetes, you not only have fasting hyperglycemia, which means that your blood glucose level will be elevated even at the fasting state, but then when you have a meal, when you're in the fed state, you have massive great postprandial glucose spikes. They go much, the increase in the glucose concentration is much bigger than the increase you'd get in a healthy person, and then the spikes are much, much broader. It takes much, much longer to bring it back down to the previous level. Okay, so those are the two things that you have, fasting hyperglycemia and enormous, very tall and very broad postprandial glucose spikes. Okay, right, and this is not good basically. Having too high blood glucose chronically like this can cause a lot of damage to parts of the body, okay, and one of the places it can cause damage is in the retina. Okay, now, the exact pathways of diabetic retinopathy are not particularly well understood. It's quite fragmented. However, what seems to happen is for some reason in the retina of people with this poorly controlled blood glucose level, you end up getting elevated levels of vascular endothelial growth factor A. Okay, now before I continue the story on from there, let's just go over a bit of basic anatomy of the retina, and we're only going to do this in a tiny little bit of detail. Okay, so we're not going to go into the detailed anatomy of the retina. All I'm going to show you is the basic layers. Okay, so in orange here, this orange layer will represent the actual retina itself. Okay, so this contains the photoreceptors, the bipolar neurons, the ganglion cells, all of that. They're all represented by this orange layer. So this represents the retina itself. Now what I want to do is show you where the retinal vessels are. Basically, they are sitting inside of the actual retina. They're sitting on top of the retina. So here would be the pupil over here where the light's coming in, and the vessels are actually on top of the retina. And the reason you don't usually see the blood vessels over your retina is because your brain has just learned to filter them out of the picture, basically. Okay, so in red here, this will represent the layer of retinal blood vessels here that's sitting on top of the retina. Okay, so these are the retinal vessels, okay, sitting on top of the retina. And then on top of the retinal vessels, you then have a membrane called the internal limiting membrane, which basically separates the retina and the retinal vessels from the vitreous humor, which is inside of that. Okay, so this blue layer here, this is representing the internal limiting membrane, and this, as I say, lines the retina with the blood vessels on top of it and separates it from the vitreous humor. So this is the internal limiting membrane. And then finally, the portion in the middle, and if you've ever done dissection of the eye, you'll know exactly what I'm talking about now. There's a sort of jelly structure in the middle that fills the cavity, basically. Okay, and this is what's known as the vitreal humor, or the vitreous humor, rather. Okay, so this is the vitreous humor. Okay, and it's not really a fluid, it's more sort of a jelly. Okay. Alright, so that's some basic eye anatomy, basically, and that's all we're going to need to understand in order to discuss diabetic retinopathy. Okay, so basically the fundamental thing that seems to happen, and it's not very well understood why this happens, is in the retina, particularly in the vitreous humor, you seem to end up with lots of vascular endothelial growth factor A being produced. Okay, so something about having this hyperglycemic state seems to trigger the production of vascular endothelial growth factor A all over the place in the retina and in the vitreous humor, etc. Okay, so you end up with lots of vascular endothelial growth factor A, and this is now going to affect the retinal vessels. Okay, now there are two different severities of diabetic retinopathy. Okay, the less severe form of diabetic retinopathy is what's known as non-proliferative diabetic retinopathy. Okay, and the more severe form is what's known as proliferative diabetic retinopathy. Okay, now they both result because of the elevated levels of vascular endothelial growth factor A, but if you've got a low level of vascular endothelial growth factor A, it will cause non-proliferative diabetic retinopathy. If you've got a very high level, it will then drive the proliferative diabetic retinopathy. So let me describe what happens in both of these. So in the non-proliferative diabetic retinopathy, you don't have enough vascular endothelial growth factor A to actually drive angiogenesis. Instead, all it does is that first portion, remember. Okay, so remember when we were discussing angiogenesis, the angiogenic pathways. Is this the portion I want? Yes. Okay, one of the first changes that the vascular endothelial growth factor A produces within cells is it increases the expression of eNOS in the endothelial cells, and it also causes endothelial cell contraction. Okay, the increased production of eNOS leads to increased production of nitric oxide, which causes the vasodilatation of the blood vessels. Okay, and the endothelial cell contraction then opens up gaps between endothelial cells and allows fluid to move out from the bloodstream into the interstitial fluid. Okay, so that's what happens in non-proliferative diabetic retinopathy. Basically, all of the retinal vessels become really leaky, and you start getting fluid accumulating on the surface of the retina, basically. Whoops. Excuse me a second. There we go. Right. So in non-proliferative diabetic retinopathy, you get leakiness of these retinal blood vessels, which then forms an exudate basically on the surface of the retina, and this causes what is known as macular edema. Okay, now let me explain what macular edema means. So the macula refers to a portion of the retina that you can see if you look into someone's eye with an ophthalmoscope. So basically, if you look at the retina with an ophthalmoscope, this is what you end up seeing. Okay, so we're looking into someone's eye, we're looking at the back of their eye, basically their retina, and basically what you find is that there is a sort of circular portion here that appears a yellowy sort of colour, like so. And this is what's known as the macula, or more fully, the macula lutea. So macula means spot, okay, lutea means yellow. Often people just call it the macula, but in full it should be the macula lutea. Okay, and this portion contains a very special area called the fovea. Okay, and the fovea is the portion that has the greatest visual acuity. It can see in the most detail. Okay, so if we want to look at something in detail, if we want to see the detail of something, we focus our fovea on that area so that the fovea is processing the visual information coming from that area, okay, so that we can see it in the most detail. That's the area that has the greatest visual acuity, and the reason is that every single one of the photoreceptors there has its own neuron, basically, whereas in other portions of the retina, you have huge convergence, loads of photoreceptors all innervating the same neuron, and therefore you lose spatial information, basically, you lose resolution, whereas here you have very, very high resolution. Okay, so the fovea is a very important part for being able to see in detail. And basically in macular edema you end up with an edema over the macula and therefore over the fovea, and this can hugely blur your vision basically, okay, as well as actually damaging the retina underneath. So that's what occurs in non-proliferative diabetic retinopathy. You get macular edema because of the leakiness of these retinal vessels induced by the elevated levels of vascular endothelial growth factor A. Now, in proliferative diabetic retinopathy, unfortunately, it's worse. Okay, in proliferative diabetic retinopathy, the vascular endothelial growth factor A that you've got is high enough to actually induce angiogenesis of the retinal vessels. Okay, so they start sprouting new vessels in the way that we've seen, and basically these vessels start heading towards the vitreous humor. Okay, they want to go to the vitreous humor, and therefore they actually start penetrating the internal limiting membrane, and they sit in this potential plane in between the back of the vitreous humor and the internal limiting membrane. So basically what you start to form is this layer of blood vessels that is in between the back of the vitreous humor and the internal limiting membrane, and you should not have a layer of blood vessels there. Okay, when you get a layer of blood vessels in that potential space between the internal limiting membrane and the vitreous humor, that's what's known as a neovascular membrane. Okay, so the retinal vessels start sending capillary sprouts through the internal limiting membrane, and they start forming this neovascular membrane in between the internal limiting membrane and the vitreous humor in front. Okay, right, now why is this so dangerous? Well, what can happen basically is the vitreous humor can end up detaching from the internal limiting membrane, and when it does, it creates a real space there, and what tends to then happen is these neovascular membrane blood vessels end up tearing open when the vitreous humour detaches, okay, and it causes massive hemorrhage basically. So what this can lead to basically is posterior vitreal detachment, and when that occurs you get massive hemorrhage of this neovascular membrane, and therefore you end up with blood covering your entire retina, and this can lead to huge damage of the retina and blindness basically. So posterior vitreal detachment, okay, which then leads to hemorrhage and huge damage to the retina and blindness basically. Okay, right, so that's the more severe form. That's where the vascular endothelial growth factor A is strong enough to actually produce angiogenesis basically. Okay, right, so clearly this is caused by this too high level of vascular endothelial growth factor A, and I'll stress again that the link between the hyperglycemia and the elevated level of vascular endothelial growth factor A is poorly understood. So if you're feeling that this is quite fragmented, it's because it is fragmented. We don't know the link between why having this hyperglycemia causes the elevated VEGF-A. Okay, but it seems to. Right, so clearly this pathology is driven then by the elevated VEGF-A present in the retina and in the vitreous humour. Okay, so if we gave bevacizumab, this drug that we have, which can just mop up VEGF-A, then we could stop this basically. Okay, and indeed we do. We now do use bevacizumab to treat diabetic retinopathy. Okay, so it's given by an intravitreal injection, which basically means that you inject it into the vitreous humour. Okay, and it works very, very well. Okay, it can prevent macular edema because the VEGF-A is underlying the leakiness of the retinal blood vessels, and therefore if you get rid of that, the edema can be cleared, and therefore it can help with the macular edema in non-proliferative diabetic retinopathy, and it's also capable of halting angiogenesis in proliferative diabetic retinopathy, and therefore stopping the progression of this neovascular membrane. Okay, so that's bevacizumab used by intravitreal injection to treat diabetic retinopathy. Okay, right. So bevacizumab is this fantastic anti-angiogenic drug that has now got a place in treating cancer and in treating diabetic retinopathy. Okay, what we're now going to talk about is the opposite case, where we want to induce angiogenesis as part of our therapy. Okay, and this is not yet realised in clinical medicine. Okay, we can't do this yet, basically, but it's a hope for what lies ahead in the future. Okay, so I'm going to talk about the potential use of angiotherapy, pro-angiotherapy this time, for the treatment of coronary heart disease. Okay, so let me tell you a little bit about coronary heart disease firstly. Okay, so in coronary heart disease, what you basically get is atherosclerosis in your coronary arteries, okay, or in a coronary artery. And this occludes blood flow to a certain portion of the heart. So let me draw you a picture of the heart here. Okay, right, so we'll have the right atrium here. Okay, and this is now going into the right ventricle, obviously. So here's the boundary between the right atrium up here, here's the right ventricle, then it will come out into the pulmonary trunk, which we'll have coming off up here. Okay, then we'll have the left ventricle here, coming into the left atrium up here. Okay, like so, and then you'll have the aorta sort of coming out from behind here, so this connects up, it's behind the right ventricle here, and it will have the aorta coming out up here. Okay, like so. So let me colour in a few of these vessels, and then hopefully this will become more apparent. Okay, so this big vessel here, which I'm now colouring in red, that's the continuation of the left ventricle down here. The left ventricle goes behind the right ventricle here, and then has the aorta coming out of it. Okay, so this is the aorta over here. This is the pulmonary trunk here coming out of the right ventricle at the front here. Okay, and this will be carrying deoxygenated blood, so I will colour that in blue. Okay, like so. And this here is the right ventricle. I'll put left ventricle here. This is the right atrium here, and this little bit peeking out from behind there is supposed to be representing the left atrium. Okay, so let me show you the two major coronary arteries. Now, I'm not going to go through all the anatomy of the major arteries of the heart, I'm just going to show you the two biggies. Okay, so the two major coronary arteries are the ones which come directly off the aorta. Okay, one of them comes off here and moves down this crack between the right atrium and the right ventricle, and that's obviously the right coronary artery. Okay, so this is called the right coronary artery here. And then you also have the left coronary artery, which moves off from the aorta behind the pulmonary trunk and then comes down this gap between the right ventricle and the left ventricle. So this is the right coronary artery, and then we will see the left coronary artery here, but it's coming from behind the pulmonary trunk. Again, it's coming off the aorta. Okay, and in vivid purple there, that is our left coronary artery. Okay, so basically in coronary heart disease, you end up with atherosclerosis, usually in one of your coronary arteries itself, or maybe a major branch coming off one of these. Okay, but let's say you end up with atherosclerosis in your left coronary artery. Okay, and this is going to include blood from flowing through the left coronary artery. So all of the tissue that this left coronary artery now supplies is going to be becoming gradually more and more ischemic, basically. It's going to be getting less and less blood, okay? And this leads to angina pectoris, which is when you start exercising. Well, when you've got this and you start exercising, obviously your heart can no longer cope, because the heart will have to be doing more pumping when you're exercising, and with this reduced blood flow it just can't handle it, basically, and you get intense pain, intense crushing chest pain, and that's angina pectoris, okay, which is basically your heart saying you're going to have to stop now, otherwise we are literally going to die because of lack of oxygen and too much demand on us. Okay, and then it can lead to heart attack, which is where the cells actually die because they're receiving too little blood flow. Okay, right. So the idea then behind how we could use angiogenesis to treat this is that if you think about this, the right coronary artery here will have branches coming off it, and they'll be giving blood to all these sort of little bits of the heart here. And then you'll have branches coming off the left coronary artery here, which will also be giving blood to these portions of the tissue as well. Okay, so the idea is that if we could promote angiogenesis, we could promote connections between branches from the right coronary artery and branches from the left coronary artery. And the idea now is that these little branches that we're forming here, these are called collaterals, basically, and they mean that now if your left coronary artery suddenly does become blocked by an atherosclerotic plaque, okay, then you're going to be getting blood from the right coronary artery coming through here, okay, and that will protect the heart, because it means that now these areas which were previously just receiving blood from the left coronary artery, now they can also receive blood from the right coronary artery. So the idea is we would like to form these collaterals, these connections between the circulation from different coronary arteries, basically, here, okay, in order that you can receive blood from other coronary arteries if one of your coronary arteries has got an occlusion in it. Okay, so that's the idea, that we would like to promote angiogenesis in the heart so that we can form these collaterals so that you are effectively protected, because you're receiving blood from from both of the coronary arteries, and then if one of them gets occluded by atherosclerosis, you can still get blood from that other coronary artery. Okay, so in coronary heart disease, we would like to promote angiogenesis so that we can promote the formation of these collaterals, and therefore you will be more protected if one of your coronary arteries then does get occluded. Okay, right, it's not a reality yet, because we don't have a way of delivering vascular endothelial growth factor A just into the heart, okay, and not delivering it all to all the other places of the body, which would cause major side effects. Okay, but that is potentially something that could be available in the future for the treatment of coronary heart disease, a pro-angiogenic therapy which establishes these collaterals which connect different circulatory systems together so that areas of the heart have blood coming from different coronary arteries, basically, so that if one gets blocked, then it's still going to have a blood supply. Okay, right, so that now concludes our discussion of angiogenesis and angiotherapy.